Hi, welcome to week 3, chapter 3, cell structure and function. This is going to be a fun chapter, at least I hope you find some interest in it. Maybe not jump up and down for joy kind of fun, but I think it will hold quite a lot of you, hold your interest, I hope. We're going to take a look at eukaryotic cells primarily. These are cells that you would find in plants and animals, so things that we're really familiar with. Obviously, they're going to be in humans as well. We're going to talk about how cells are manufactured, what is contained within their cell walls, and the function of each organelle that resides within that cell. There's a lot to one little cell, and you'll be amazed at how much one little cell can do, and then when you multiply it by the millions of cells that we have, it's pretty amazing to see how we've evolved into the beings that we are today. So we will jump right into it. This chapter is, again, like the last chapter. It has a lot of information, and so we're just going to take it page by page, and hopefully you won't get too overwhelmed. Feel free to stop your recorder and go for a walk or drink a soda or whatever. Maybe you need a glass of wine, but don't feel bad if you have to stop and then come back to it later. Sometimes that's the best way to study. Let everything kind of gel in your mind that you've been hearing and reading about, and then come back to it later. That's always a wise choice. Okay, let's get started. Before we can talk about cells and their components within, we need to talk about the history of the microscope. So Antony Leeuwenhoek was a textile merchant. He wasn't a scientist. He was just a regular, normal man who had a very curious mind. And one day, while he was at home cleaning out his workshop, he broke a glass. It was like a glass bottle or something. And so he broke it, and when he was cleaning it up, he happened to notice how the light caught the reflection of this glass, and it actually amplified the image on the other side. There was like a piece of paper or something that he was looking at that was on one side of the glass, and he happened to be looking through the other side. And so it had this little magnification effect, and that got him really curious. And so he started taking bottles, breaking off the bottoms, and polishing them and honing them down into these little cones of glass. And he made a device, which you see here on the screen, that would hold the glass figure. It's a little bit elaborate, I suppose, but he would put a specimen on one side and the glass in the middle in that vise, and then have the sunlight shine through, and it would magnify the image. He was particularly fond of looking at water molecules. He would put water on a glass or on a container of some type and look through his little micro glass that would be in this device and see what he could find within it. Sometimes he didn't find anything in the water. It was just clear. But other times he would find little things that were swimming around or moving in some way, and as he got closer looks at these things, he called them animalcules and beasties. And these were the first specimens that had been viewed by a microscope. And over the years, he continued to work and perfect and hone his microscope till eventually we got a version somewhat similar to what we use today, only it was a single ocular glass that would fit in this machine still, or in this vise, and he would just hone that glass down and shine it and polish it so that it was really thin and really smooth, but he got the effect that he was wanting. And the more curved he had the glass, the better refraction of the light he had, and the ability to emphasize these figures even more. So So I've always found that really interesting, that somebody who wasn't a scientist still had an inquisitive mind like a scientist and gave us one of the most important tools that we've ever had. And that's how he became the father of microbiology. And I have a book I'll show you in lab that I will bring out that you guys can look at. It has a bunch of different scientists on it, and I find it really interesting. So I'll bring that so you guys can look at. Now, Robert Hooke, as you can see here, was using some magnifying glasses, some microscopes that were built upon Leeuwenhoek's idea. And so you can see here where it has a little bit more sophistication to it than his original polished glass that Leeuwenhoek's had. With these particular magnifying glasses, or microscopes if you will, Robert Hooke had looked at these samples of cork, and they were just very thin shavings of cork. And you can see here in the circular picture how thin they were. I don't remember why he was looking at cork, but maybe it was just because he could. So he's looking at this through a microscope, and he sees all the cork has these little holes all over it to make it sort of look like a lattice work or a loose blanket weave of some sort. And so he called each one of those little sections, those little squares that he was looking at, he called them cells. They reminded him of prison cells of all things back then. They were dark and square and dirty, and that's kind of what he was seeing here. And so he transferred that name cell over to this particular organism that he was looking at. In this case, it was a cork. And so Robert Hooke and Leeuwenhoek, they were early pioneers of microbiology. And from the microscope work that Leeuwenhoek had and Hooke's inquisitive mind about looking at things under those microscopes, and developing even more tools and more ways to get even in closer than what this picture shows. Those are, really important people that you need to know about as you study your scientific ideas. I think it's good to have this background, and you can always win a trivia game if nothing else. So these are just some early on advantages that we were given, which I think were pretty cool. Thankfully today we have use of very sophisticated microscopes that have come an incredibly long way from the 1700s and Leeuwenhoek and Hooke. And we have the microscopes that we use today in our lab, which are like the one that's pictured here, the dual ocular light microscope. And so, as you can see, there are tons of different kinds of microscopes, and they range from $25 that you could get for a child all the way up to millions of dollars. And most of those high dollar microscopes we probably will never see unless you work in a lab that happens to have one, or work in a hospital actually that will have some of these. The TEM, transmission electron microscope, that you'll see pictured here, and the scanning electron microscope, and some others, they have incredible detailed quality of getting really close into a specimen. I mean, it gets down to the nitty-gritty, to the point of where in one picture you'll see it looks like an alien with his little teeth sticking out, but it's so teeny tiny you would never be able to find it with the naked eye. So the ability to look at these microscopes is really incredible, and the sophistication of them have come a long way, and the use for them have come a long way. There are some that you can use where you can put in a colored specimen and it will detail all the structures in that specific color. Like certain aspects of organisms will light up in a different color or will take on stain in a different way than some other ones will. And you'll see this in lab. Sometimes when you stain a specimen with a purple dye, let's say, only certain structures are going to take up that dye, and that is going to mean something to the scientist that's looking at it. Sometimes if we see things that are lit up, that indicates a pathology. And sometimes if we see things that aren't lit up, but we still see the structure, that indicates a pathology. So it just depends on what you're looking for. But microscope work is a ton of fun. We have a lot of cool slides in lab for you to look at. And I encourage you not to miss those labs, because the foundation that you'll have will carry on throughout your other courses, including microbiology, which I encourage you to take. It's a really fun, really cool class. It's one of my favorites. So here's another depiction of just how detailed and microscopic we can really get. This is a head of a pin. And you always hear people say, well, if it could fit on the head of a pin, I would do it, or something like that. I'm probably just showing my age right there. But this is a head of a pin, and it has some bacteria on it, which is what the orange stuff is. And then if you look at the middle picture, you can see a little bit more. It's a little close up. You can see a bunch of different individual pieces of bacteria, or this possibly could be yeast. And then the last picture will be of one single or several single of the bacteria cells. And so it's incredible how close and detailed we can get. And as you can see, we have color, we have detail, we can see all the structures, and it's all thanks to specific, sophisticated microscope inventions.
By definition, a cell is the basic unit of life. It can carry out metabolism, maintain homeostasis, and reproduce. It can exist alone or in combination with other cells. Most living organisms are made up of multiple cells, and each of these cells reproduce by dividing. And when they do, they pass on their DNA, their hereditary information, down to each of their offspring. If a single cell reproduces by dividing itself, then it stands to reason that each cell has come from a pre-existing cell. This process is called the cell theory. As you can see here, there are 4 tenets of the cell theory that you should know. The cell theory states that all organisms consist of one or more cells. The cell is the smallest unit of life. Each new cell arises from another cell, and a cell passes hereditary information on to its offspring. Remember, I'm always talking about how an organism's goal is to find a mate and reproduce and pass on its hereditary information to its offspring so that they can go on, find a mate, and reproduce and pass on the hereditary information. This hereditary information being passed on is critical to the survival of every species on the planet. Without the ability to do this, the species would die off. So we always need to have this hereditary information. It's sort of a blueprint of the world as that species knows it. It's the blueprint of how that species functions within the world around it. And so we need to have this information so that we can go on and be successful beings. We've been talking in general about cells, and now this chapter 3, is going to delve a little bit more into the interior of a cell. And there are 2 types or 2 categories of cells. So remember when we talked about domain names, we talked about Dear King Philip came over from Greece singing his common name. Well, the D in that, the Dear King Philip part, dear indicates domain. And so in this textbook, we're talking about two separate domains, prokaryotes and eukaryotes. And if you remember, the prokaryotes can be further divided into bacteria and archaea classifications. And as a reminder, some textbooks that you come across will actually have those separated out into 3, sometimes even 4 separate domains, pulling out the bacteria and archaea as their own separate domains. But typically, we go by prokaryotes as a domain and eukaryotes as a domain. And eukaryotes are pretty much everything that's not a bacteria or an archaea classification. And so that means that we are talking about humans and four-legged furry friends and fish and plants and, pretty much all the little familiar animals and organisms in your day-to-day lives that you're familiar with, and including a whole bunch that you're not familiar with. But that's an easy way to think of a eukaryote. A eukaryote is you. Okay, now how about that for a reminder? Some of these, between the prokaryotes and the eukaryotes, there are some similar structures and similar components to each. So we might look at both of them and say, well, they have this component here, but hey, you know what? I found this over here as well. So prokaryotes and eukaryotes can have similar structures, and we'll get more into those in detail in just a few slides. This particular slide is showing you the area where DNA is housed. So in this particular picture, it's showing you the nucleus, which is outlined in that white outline. And the interior, all that granular looking stuff, that is your DNA. So this whole structure here is your eukaryotic DNA organelle. It's your nucleus. The nucleus is the organelle. And then the dark circle there in the lower right portion of that organelle is the nucleolus, and that's just another region of the DNA. You'll notice also on this slide that prokaryotes have a nucleoid region. And what this looks like, we'll see on another slide, is very similar to the nucleus of a eukaryotic cell, except it doesn't have a cell wall. It doesn't have a membrane around it. In eukaryotic cells, it's very important to have a membrane around the DNA, around the hereditary information, to give it an extra layer of protection. The prokaryotes don't have that extra formidable layer. It doesn't have a membrane surrounding its DNA. It's just the DNA is packed tightly in this one region, and they call that the nucleoid. So this is a typical prokaryotic body plan. And think of bacteria in this case. You can see it has this outer green structure. It looks like a pill capsule, and that is exactly what it's called. You can see this cross section here shows the capsule as the outer membrane in that light green color. If you go inward one row of green, that is next one that it's showing is the cell wall, and then the skinny one on the interior is the cell membrane. So these body structures are quite strong. We've got 3 membranes and a hard outer shell to protect us. And the interior light blue color, that's your cytoplasm. And then the squiggly stuff is your DNA, housed in a nucleoid, not in a nucleus, in a nucleoid. That makes it special and distinctive to a prokaryote. The ribosomes are the little speckled areas, and then on the outside of the capsule we have these structures that look like hair, called pili, and they help protect the capsule or body, just in another fashion. And in some cases it will help move the capsule around or the body around, but in most cases it's for protection. And then the flagellum is the long whip-like structure you see. Looks like a big tail. And the tail will actually do that. It will whip around back and forth just like a whip, if you were to crack a whip, and it will move the prokaryote, the body, forward. Now, you'll notice at the bottom the distinguishing characteristics for prokaryotes are the nucleoid and the pili. Eukaryotes have cilia instead of pili, and they look almost identical. But yet, you'll notice on the upper left that we have similarities to plant and animal cells. Plant and animal cells are types of eukaryotes, and so across the domains, these structures have some similarities. They both have DNA. They're housed differently, but they both have DNA. They have cell membranes, they have ribosomes, they have a cell wall, and in some cases in the animal cells, there are flagellum, just like in the prokaryote. So we do have some differences, or similarities between or amongst all domains, which is pretty impressive, I think. And then over time, is thought that they've become differentiated into the structures that we know today. So now we're going to talk about the interior of a cell, the diameter of the cell, in that the surface to volume ratio is very important. As the slide says, size does matter. If you have a circle, or let's actually think of a tennis ball. If you cut open the tennis ball and you saw the interior, it would look like a cup, and you could see the outer walls. And let's say that those outer walls were porous and material could pass in and out. Well, that structure has a very distinct size and diameter to it. The cells have the same type of situation. If you were to need potassium, for example, the potassium would go in through the cell wall, enter to the interior of the cell, and move through the cytoplasm to the organelle structure that it needs to carry out whatever function it's going to carry out. If the organelle is on the opposite side of the cup, let's say, or the cell, it's going to need a lot of momentum and effort to get across the entire cell. And if a cell is proportioned properly in the surface to volume ratio, then that potassium can get to where it needs to go, even if it's all the way across to the other side. If a cell was too large, the potassium would not be able to reach the other side. It would just burn out before it could even get there, because it didn't have the strength or the momentum to get all the way across to the other side. If a cell is too small, it's just simply not going to have enough room for everything. Because as you'll see later towards the end of the discussion, there are a lot of components that are within and embedded within the cell and the cell wall, respectively. And so the surface to volume ratio limits the cell size and influences cell shape, and must be small enough or appropriately sized enough to efficiently exchange materials within the environment. And then you have the definition at the bottom, which is a little too mathematical, math related for me. As you all know, I'm really great at math. So, surface to volume ratio is defined as the relationship in which the volume of an object increases with the cube of the diameter, but the surface areas increase with the square, which is a nice mathematical way to say that the surface to volume ratio limits cell size, influences cell shape, and in order to effectively and efficiently exchange materials within the environment. So focus on whichever definition makes sense to you. Components of eukaryotic cells. We'll talk just a brief moment about the plasma membrane, and then at the end of the lecture we'll talk in depth about it. But basically it is this lipid bilayer, which is just a fancy way to say that we have this fatty component that has created two layers that surround the cell. And so plasma membrane is a lipid bilayer, and it has proteins embedded in its surface, and it surrounds the cell and controls which substances move in and out of the cell. And so that means it's selectively permeable. It can keep some components out and let others in. It's very selective. I like to think of it as a bouncer at a nightclub. Some things will be let in and other things will not be let in. And so if you look at the interior of a cell, you have this substance. It's called the cytoplasm. It's semi-liquid, and it's just dense enough to hold all the organelles within the cell in place so that they're not all floating around all willy-nilly. Because, for example, if that potassium that we were talking about earlier were to enter into the cell and it needs to get halfway across the cell to the specific organelle that it needs, if that organelle is not where it's supposed to be because it's floating all around, then the potassium probably won't reach it, and then that function that needed potassium won't be carried out. And so the cytoplasm is this semi-fluid substance, and I like to think of it as hair gel, and you'll see why in just a second. But it's just fluid enough so that things can move that need to move, but hold organelles in place. And I think that's important to note. And you'll notice on the next slide why I call it hair gel. So here are 2 jars of hair gel. And as noted in the previous slide, I like to compare cytoplasm with hair gel. And to me, if these were cells, we have a red cell and a black cell. This is what a cell would look like, even though they're not. These jars are not perfectly cylindrical or round. Spherical is the word I was looking for. If they're not totally spherical, but the gel inside them, you can see the bubbles are suspended within the gel substance, and you can think of those bubbles as organelles, and that's how the organelles work inside the cytoplasm. They have this suspension to them. They're suspended in this hair gel, or actually in the cytoplasm. And it should be noted that cytoplasm technically is the fluid portion and the organelles combined. The fluid portion alone, if you took out all the organelles, the fluid that's left is just called cytosol. So cytosol and organelles equal cytoplasm. But we always say cytoplasm to mean this fluid portion. So you may hear that word cytosol, and that's what that indicates. But I really think that the hair gel does a nice depiction of what cytoplasm is. And now you've heard me talk about organelles being suspended inside the cytoplasm, and organelles are just structures that carry out specialized metabolic functions that are within the interior of the cell. And so basically, everything but the nucleus are organelles. And so the nucleus is there in the middle. It's that gobstopper round looking structure. It's cut open so you can see the DNA within, which is the purple portion. It has black dots on the outside, which indicate the nuclear pores. And then it has these ribbon-like structures. Some are coming straight off of the nucleus, and others are completely separate from it, and those are all called organelles that we'll talk about individually in just a little bit. The blue portion of this picture on the interior of the cell, that is your cytoplasm, or hair gel, if you'd like to think of it like that. And if you notice on the outer part of this cell, the dark blue portion has some water droplet like things coming off of it. That's just depicting that it's a semi-permeable membrane that allows materials to pass in and out of it. This is just showing you what cilia looks like. If you remember on the prokaryote body plan, there were the pili or the pilus that were coming off the outer capsule of the body, and it looked like little hair-like projections. Well, this is the same thing as cilia, only on a eukaryotic cell. They're hair-like structures that project from the membrane of eukaryotic cells. They have coordinated beating fluid that propel them forward or around in various motion, and it provides some protection as well. This is showing you what the flagella on a eukaryotic cell would look like. This one happens to be on a sperm. It propels it through fluid. It does that whip-like motion just like it does on the prokaryotic body plan. They're both called the same thing. They're both flagellum. These are called pseudopods or false feet. These are the arm-like structures that are extendable lobes of membrane enclosed cytoplasm for movement or to engulf prey. And hopefully we'll be seeing these in lab this week. They're very cool to see. They're amoebas, and the cytosol within them will propel those arms forward, and as material that needs to be ingested comes near the amoeba, the arms will shoot out and surround it, and then eventually pull the food inward towards that center mass, which is the darker portion in the center. And so these arms or false feet are called pseudopods, and these are for eukaryotic cells as well. Okay, on this slide, you're going to see pretty much all the important parts of the interior workings of a cell. This is a eukaryotic cell, and we'll just go through it number by number. I think it's the easiest way. I really like this picture. Again, it's one of these cartoony type pictures, but it shows everything really well. So number one is the green structure that you see in the upper left. That is your nucleus, and it has actually cross sections, so you can see the interior, that little bit of the pale granular looking purple stuff. That is your DNA. So that is housed inside the nucleus. The number two structure on the outside, the green little divots that you see, those are actually holes. They're called nuclear pores, and they allow material in and out of the cell. much like the lipid bilayer of the cell membrane. Number 3, that is the endoplasmic reticulum, and so is number 4 down in the lower left. One is called rough, so number 3 is called rough, and that is because it has these structures that are numbered 5 called ribosomes, and they are on the rough endoplasmic reticulum. They will have to do with protein synthesis later on. We'll talk about that. But for now, the endoplasmic reticulum is actually a structure that's sort of ribbon-like, and it's got these twists and turns, as you can see, that come off the nucleus and are have these little dot-type structures called the ribosomes. The endoplasmic reticulum that's in the lower left does not have ribosomes on the outside. The one thing I don't like about this picture is that it does show ribosomes on the interior components of the smooth endoplasmic reticulum in this lower left area, but you'll notice that the ribosomes do not appear on the outer folds of the endoplasmic reticulum, so this particular portion of it is called the smooth endoplasmic reticulum. So the rough has the ribosomes, it's the larger ribbon-like structure coming off the nucleus, and then the smooth endoplasmic reticulum is the shorter, smaller ribbon-like structure without ribosomes on the exterior of the folds. Okay, and so again we said number 5 were the actual ribosomes. Number 6 are proteins that are going to be transported, and they're going to be using number 7, which are transport vesicles, and there's several different kinds in here, but these are structures that help proteins move from the endoplasmic reticulum all the way out into the exterior of the cell. All right, number 7, again, these little round circles, these are called vesicles. Sometimes you'll see them cut in half and they're little cup-like structures. Number 8, don't get this one confused with number 3 and 4 with the endoplasmic reticulum. Yes, it's ribbon-like structure, similar. There are some ribosomes passing through it, along with some transport proteins and some fatty acids you'll see, and lipids and things. But the Golgi apparatus is completely separate. You'll notice that you could be able to differentiate it from the endoplasmic reticulums because it doesn't attach to the nucleus like the endoplasmic reticulums do. Okay, so it is a ribbon-like structure. It's a little bit smaller. It's usually depicted in a different color in these in most photos, other than the live photos of what you might see. I think we show you one on the next slide. But they're detached from the actual nucleus. Now you see some other numbers in here. We're not going to worry about what every little fold and curvature on the Golgi body is going to look like or act like, but just know that this whole structure here from 8 through 11 has to do with the Golgi body, or as it's called here, the Golgi apparatus. Here's a picture. It's a little difficult to see what's going on here, but this is depicting the Golgi body, and then these round circular structures are vacuoles. Okay, so if you look in the lower portion of the picture, it looks like this ribbons or string, these lines that go back and forth in sort of a smiley face. That is your Golgi body. And as the slide says, this is the cell's post office. This is where materials will be gathered, and polypeptides and lipids will work to sort and package finished products and transport them into the vesicles. And then the vesicles can take them out to the interstitial fluid. The vacuole are holes, basically. They're fluid-filled organelles that isolate or dispose of waste, debris, or toxic materials. So that's why there's so many of them, because you don't want to transport waste all the way across if you don't have to. That's a waste of energy. And so we'll just produce waste and throw it in the nearest garbage bin, which is the vacuole closest to us, and it will get rid of the waste there. Even though the cell to surface volume ratio is adequate and materials could go all the way across the cell to be disposed of, you might as well just throw it right outside your back door, right? Instead of running down to the local dump. So the vacuoles are the white circular oblong circles here, or some of them are a little bit grayer, but they're the circle looking structures. Those are your vacuoles. This slide is talking about mitochondria and chloroplasts. Chloroplasts are what are pictured here, and these little green structures that you see, these are chloroplasts, and we will talk in detail about these in the next chapter. But they are a type of organelle, and it's a membrane enclosed organelle for photosynthesis. So that's what a chloroplast is. We'll talk more about that later. The mitochondria, which are not depicted here, are a unique structure. They have their own DNA, and they resemble bacteria that may have evolved by endosymbiosis. And so what that means is that a long time ago, and by long I mean like 100 million years ago, there were these structures or organisms that looked like bacteria that were once thought to be bacteria, that were engulfed by another organism. And the two organisms together had this symbiotic relationship. And it says endosymbiosis because the mitochondria were housed within this other structure. And over time, they ended up with a symbiotic relationship, sort of you wash your back and I'll wash mine. Actually, I'll wash your back and you'll wash mine. Is that how that's supposed to go, right? Anyway, you get the idea that they are helping each other. And so over time, with evolution, the mitochondria just became a regular component, an organelle, if you will, of a eukaryotic cell. And so you'll see these depicted in the next slide. So this is what mitochondria looks like. This is that ancient structure that was once housed within a cell and had this symbiotic relationship with it millions of years ago. And through evolution and time has become a permanent structure of eukaryotic cells that we now no longer can do without. And so you don't really need to know the components of this mitochondria. Just want you to get an idea of what it looks like. So if I pointed to one on a test, you would be able to recognize it. Okay, so this is what it looks like. Before I get into this slide, I wanted to redirect you back to that figure 3.7 that I mentioned earlier under the art connection in your textbook. The two eukaryotic cells here that are pictured, the animal and the plant cells, show all of the interior organelles that each cell houses. And you'll see repetition here that they both have Golgi apparatuses, or apparati, I think it's called, the mitochondria and the vacuoles and the peroxisomes and lysosomes. microfilaments, which we're going to talk about in a second. It gives you a little bit more detail here, so it's good to take a look at that and read through the description of each organelle that it gives you. Okay. Now, your cytoskeletal elements. These are components that will give the cell structure, among other things. It's called a cytoskeleton, and it's found between the plasma membrane and the nucleus. It's made up of protein filaments that are tightly bound and aid in reinforcing, organizing, and moving cell structures, and sometimes even moving the cell. So this is another form of transport, but on a very minor scale. Okay, and so that's what you're seeing here in these two pictures. Microtubules are made from a protein called tubulin, 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 and they grow in a specific barrel-shaped pattern like this yellow one. They work in supporting various cell activities, such as in-cell division. They will have a function in that. They're like little soldiers who take up formation, and when duty calls, they go over and they do their job, and when they're complete, they go back to rest. So they have to be called to action, basically. Microfilaments are the fibers that are made of a protein called actin, which are the blue structures here. They work to change the shape of a cell, and they form in a mesh pattern to provide cell strength. So if you have ever heard of like a bladder sling or a hernia patch, these are meshes that hold body structures together in place. It supports a bladder, for example, or it provides rigidity against like an intestine, in a hernia type of situation. And so it's this mesh, just like a screen. If you were to look out your window through a screen, that's what the mesh looks like. It's similar to that kind of pattern. And the microfilaments here can cause the cell to contract as needed. There's intermediate filaments as well that provide support to the cell, and they're the most stable parts of the cytoskeleton. So those are the ones here that are talking about where the elements that lock the cells and tissues together. It's really giving that nice reinforcement here. Actin, you might also hear about later, is found in muscle cells, and they will have a part in contracting and releasing muscles. So obviously on a much larger scale, because muscles have are quite large, and they require a lot of cells for their structure and to make the movement of contracting and releasing. Remember when we talked about a single cell, we said that the cell membrane is semi-permeable to let in and let out various elements, right? Well, that was one single cell. Well, here you can see when you have several cells put together, they are not glued together. They have a area between them that you can see, especially if you look at the top of this structure that it's showing. You can see that there are spaces, and the spaces of each cell, each cell, I should say, sits within an exterior liquid called the extracellular fluid, or in medicine we often call it interstitial fluid or extracellular fluid. So each cell sits in that. But here, if we're talking about multiple cells, because as we know, multiple cells can make up tissues, and then tissues can make up organs, and so on. Here in this picture, you have a grouping of cells that are probably forming a tissue of some sort. And so when you have a grouping of cells, depending on what that grouping is for, will determine what type of junction connects each cell to each other. And so we've got several different types of junctions. Here we're talking about the first type. It's called a tight junction, which is where these fibrous proteins join the epithelial cells. The epithelial means skin, so it's joining skin cells here. It's creating actual tissue, and it's preventing fluids from leaking between them. And so think about that for a minute. If you look down at your arm, you're going to see your skin. If we were able to look interiorly to our skin, we would see all of the cells and the muscles and things like that. But we would be able to see the fluid that all of these cells sit within. And so if you have fluid under your skin as part of your normal makeup, because that's what's holding all of your cells together, basically. If these cells were not tightly bound, that liquid would just be oozing right out of your arm, right? And so sometimes, like if you cut yourself a little bit and you have this oozing nature happen aside from blood, that is what that is. That's interstitial fluid, and it's oozing out of you because these tight junctions have been broken. And so this particular junction will hold epithelial cells together to prevent that fluid from leaking between them. Here's another type of adhering junction. These particular ones are anchors that will hold each cell together. And it says here, anchors cells to each other or to the extracellular matrix. And so it's got these finger-like projections. So if you stuck your fingers through a piece of cloth and grabbed onto something on the other side and pulled whatever you grabbed onto close to the fabric, you've created this junction where these finger-like projections are holding tightly onto something on the other side. So in this case, it's holding onto each other. It's showing you can see how it's got this matrix in between the two cells, and then the finger-like projections are allowing the matrix to be held in place. So if those were to be bent over in some way, they can't be pulled back through to the interior between the cells. This is the last type of junction between cells. This is called a gap junction. It forms a channel across plasma membranes of adjoining animal cells. And so you can see they've done a cross section here, which is the edge of this blown up cells that are here. You can see a channel going east to west or left to right in the green junction, I guess you would call it. So it's got this little channel happening, and then if you look on the exterior of each cell, you can see the round green flower-like structure. That's just the end of the tubule that has end of the channel, I guess you could say. This is allowing materials to pass between the two easily, but not get trapped in between cells. So it's going through one cell across the junction into the other cell, and it will transport materials back and forth that way. And again, it just keeps materials from falling in between the two cells, so it's bypassing the interstitial fluid, essentially is what that's doing. So you've got That's your third kind of junction. Earlier in this lecture, we talked about the lipid bilayers that are make up the membrane that surrounds a cell. And again, we're talking about eukaryotic cells here. This is a nice depiction of what they look like. So here you have your round cell that we all think of, and then you can see the bottom cup, if you will. The interior of it has that liquid makeup, that cytoplasm or cytosol. Cytosol just means that it's the liquid portion without the organelles in it. Cytoplasm includes everything, but again, it's that gel-like structure. So think of hair gel again. But what this is mostly focusing on is the outer cell membrane and what it's made of. And so you can see the edge here. It's pointing to several different things. It talks about the hydrophobic tails, which are the interior of that top ring, and the hydrophilic heads, which are on the outer portion and then the interior portion of this ring or membrane. And so they say the tails are hydrophobic, and that's why they point interiorly toward one another, because they need to be furthest away from the fluid inside the cell and the fluid outside of the cell. And so they're kind of phobic. They have this fear of water or fear of liquid, so they're going to point in towards each other, and then the heads are going to be the protector of those tails. The heads are going to form this barrier so that the liquid doesn't get to the hydrophobic tails. So that's why we have heads on the interior where it would be bathed, I guess you would say, in the cytoplasm or the interior fluid portion of the cell, and then the other head on the outermost portion is going to be bathed in the interstitial fluid or the extracellular fluid. And so those are the heads, and they don't mind being in the water because they're water loving. That's what philic means. Philic means loving, and hydro means water. So these are hydrophilic heads, and then the hydrophobic tails are the interior part. And so this creates the lipid bilayer of the plasma membrane that surround each cell. This picture is more typical of the way you will see the phospholipid bilayer depicted. It's more cartoon-like, but even though we talk about the cell as being a circle, it's easier to picture the lipid bilayer as a rectangle like this with horizontal layers instead of the full complete circle. And so if you notice, you can see that there's the top row and the bottom row. We've got one layer of phospholipids on top and one layer on the bottom. And it's easier to notice that on the right hand side instead of the left. The left hand side, the little brackets are just a hair off of what they should be, but you get the idea. And so the red parts, they look like matchsticks. That's the head, the hydrophilic head. And so you can see there's one at the top and one at the bottom. And so we'll say that the bottom portion is facing the interior of the cell and the top portion is facing the interstitial fluid. And then the lighter French fry looking legs are are the hydrophobic tails. And so again, remember hydrophobic, it's afraid of the water, so it can't be near the interstitial fluid on the outside or the cytoplasm on the inside. And so again, this is just another depiction of that. And together they call this the fluid mosaic model of the cell membrane. And I have a good picture of that on the next slide.
Between the living machinery of the inner cell and the harsh conditions of the outside world stands the cell's plasma membrane. As crucial as this barrier is, it's surprisingly flexible. Push it and watch it move. Poke hard enough and it might break and begin to regroup. The lipid molecules of the membrane naturally assemble in a double layer because their tails repel water as their heads attract it. Throw in some cholesterol and a few carbohydrates, and you have the basic structure of a plasma membrane. Within these lipid molecules, we also find different proteins which do various things for the cell. For instance, they receive signals from the world outside, or they transport nutrients and waste. So nature composes the membrane with a combination or mosaic of different lipids, carbohydrates, and proteins. And these molecules are not stationary. They constantly move within the structure, fluidly changing their positions. The survival of all life rests on this veil of material. A supple membrane, just 2 molecules thick.
This picture is in your book, and I like it. I think it makes a lot of sense. So you can see, even though it's a cartoon, you can see that the red heads are your hydrophobic heads, or excuse me, hydrophilic heads, and then your interior hydrophobic tails. And so you've got these little, they look like little men all lined up together, don't they? And so you can see the bottom ones are the intracellular matrix or the cytosol, cytoplasm, and then the upper ones are facing the extracellular fluid or extracellular matrix, as this whole thing is called. And all the other squiggly things in between, all the blue, I guess you'd say rectangular components, and there's some squiggly blue things and some Y-shaped green components, all different kinds of things. These are all membrane proteins, and there are a handful of types, and they are what aid in transport of materials across the membrane. So things that can come from the extracellular matrix go through, for example, if you look at the blue structure on the far left, it looks like it's got a channel in it. Material will come from the extracellular matrix side. It will fall right into that channel and slip right through without having to bother those lipid portions, the heads or the tails, and it will sort of bypass all of that, and the material will slip right through into the interior of the cell and be used for whatever it needs to be used for. If you look at the similar looking structure in the center of the picture that goes through the matrix, there is no channel there. It looks like it's closed, and that's exactly what it is. And same with the little blue one in between the two. In order for these to open up, they do have a channel in them, but they have to be sweet talked into opening up. And the way we do that is if we are opening up from the outside, from the extracellular matrix side, and bringing materials to the interior of the cell, we're going to have triggers that will open that channel up. And a lot of times that trigger will come actually from the interior of the cell. And so let's say we have potassium that's trying to get into the interior of the cell. The potassium will come up to the blue protein. Let's say it kind of knocks on the door. The protein recognizes it as something that is okay to go into the interior, but it has to just double check and make sure. So it asks the doorman on the other side, on the interior intracellular matrix side, and that doorman says, yeah, I think it's okay to open up, but I'm going to need some energy to help me do that. And so it will take energy in the form of ATP, which we will talk about in depth another time. The energy will be used to basically slide open this protein to have the channel exposed that will then allow the protein or the potassium to come into the interior of the cell without having to go through all the extracellular matrix and the intracellular matrix and those hydrophilic heads and the hydrophobic tails. It's kind of a lot to go through. And so instead, we'll just come up and knock on the door. And that's how a lot of these things work. And then it works vice versa as well, from things trying to go from the interior of the cell to the exterior of the cell. And so this is a pretty good depiction of that, even though it's a cartoon.